Hi, I'm Matthew Schumm, and in this video, I'll be talking about building a mechanistic understanding of mussel bed boundary formation. Mussels are bivalve mollusks found on rocky shores around the world, and they exhibit discrete vertical zonation, and my model may be able to help us understand why. I'll also be talking more generally about abrupt state changes, how they are produced, and whether they can be reproduced from continuous gradients in biotic and abiotic factors. So, continuous gradients might lead to nonlinear responses and abrupt transitions when a system exhibits positive feedback. So in this figure from Kefi et al., we see an example of how positive feedback can act to create abrupt transitions and bistability. So some external condition, in the case of mussel beds, it might be percentage time submerged. In Kefi's example, its amount of rainfall will vary continuously, but there is positive feedback in the system's response. So in this case, there's positive feedback between vegetation cover, which is the ecosystem state, and water held in the soil, this abiotic factor. So this leads to an abrupt transition and even the possibility of hysteresis in the transition to a new stable state. So we can extend this to thinking about mussel beds, and specifically for the purposes of this model, beds of California mussels in the Pacific Northwest. So mussels require exposure to water for food, respiration, and for protection from fluctuations in air temperature. They grow on rocky shores at depths where they are submerged for at least 5 to 10% of the time. Below this, they can grow at any depths, and the deeper they are, the larger they can grow. But the growth downward of mussel beds is constrained by Pisaster seastar predation. Therefore, we have a continuously varying environmental condition in seastar predation, which drops off higher up where the bed is less submerged. We also have the potential for positive feedback in maintaining a mussel cover state, because sea stars can only eat mussels that are so much bigger than their own body size, and so a small mussel surrounded by large neighbor mussels might be protected from predation. So here's an illustration of all the dynamical rates that we need to think about when modeling this system: mussel and sea star entry and exit, mussel growth, and sea star predation. The environment being modeled in NetLogo 3D is a section of a very gently sloping rock slope ranging from about 1 meter above the mean lower low tide, where mussels are submerged over 60% of the time, up to about 2.4 meters above the mean lower low tide mark, where mussels are submerged less than 10% of the time. The rock slope is modeled as the floor of my 3D space, with the mussels attached to it. Because this is NetLogo, the modeling environment is split up into discrete cells, called patches in the NetLogo lingo, that I use in parameterizing some of my effects, but all the agents exist in continuous space and are not locked to the center of cells. So here's an overview of the model schedule. At each time step, mussels may move around in the space, and I'll go more into detail later on how this is parameterized. New mussels then recruit into the space with some probability. Sea stars then move with some probability given their current depth and whatever randomly generated direction they're facing. Sea stars then have a chance to eat mussels at the maximum predation rate. They eat one per day. Mussels then grow a deterministic amount set by tide height. They potentially experience mortality, and then disturbances may occur that rip up sections of the mussel bed. So now here's some more detail on the parameterization of the different classes of individuals. Mussels grow according to a von Bertalanffy growth function with an asymptotic size set by their current depth, i.e. the proportion of time they are submerged. All variation in individuals in the model is assumed to be due to environmental variation. There's no parameterization of differences in individuals' genetic backgrounds. Mussels follow an exponential survivorship model with probability of dying at any time step set by a constant mean time to death, which depends on depth. Mussels can only recruit to rock that's assumed to already be covered by algae or mussels or some other biotic substrate that mussels will recruit to, as they don't generally recruit to bare rock. Consequently, they don't recruit to cells that have had mussels recently removed by disturbance. Mussels can be pushed downslope by larger mussels, and larger mussels may suddenly detach and move to other sites elsewhere on the slope. Mussels that have moved downward will grow rapidly because they have a new L-inf. Mussels that move upward are jerry-rigged to stop growing if their current length already exceeds the predicted size for them at that new, less submerged depth. And then, owing to this being a 3D model, we can have mussels recruiting on top of other mussels. So moving on to sea stars, at each time step, sea stars are facing in a certain direction. At the bottom of the mussel bed, the lowest depth where they're most submerged, they have a 50% chance of moving regardless of the direction they're pointed in. This varies across depth until at the top of the mussel bed, they're guaranteed to move down and guaranteed to not move further up. Sea stars have a certain prey size range based on their own body size. Sea stars enter the model randomly according to a Poisson distribution based on what Robles et al. Used in their sea star mussel modeling paper in 2010. They enter and exit at the margins of the model, and they won't leave the model if they've recently consumed food, which they can do at every time step. Disturbances are also a separate class of agents in the model. Each cell has a probability of a disturbance being introduced, and disturbances take radii randomly from a log-normal distribution. Every cell within the radius of the focal disturbed cell loses any muscles that it has. Every cell with muscles removed is assigned a time to recovery of recruitability that is a function of the radius of the disturbance and the distance of the disturbed cell from the focal cell. So, once model output is generated, it is processed in R. The R-packaged landscape metrics is used to calculate the clumpy index of aggregation intensity, which varies from no aggregation at -1 to fully clumped at 1 from the raster data. We also compare our model output to photo data, where we score cells with 25% of cell area taken up by muscle as having adult muscles present, as opposed to absent. The clumpy metric we use only analyzes cover classes separately. Consequently, it's robust to variance in proportion covered of a landscape, but clumpy is biased by environment size. So the sampling windows or boxes used for the output data are standardized to the size of field photograph sampling grids, so boundary intensity can be compared. This is because all of the field photos are of 50 centimeter by 50 centimeter quadrats. The boundary level of tide height around which clumpy is calculated for the sampling grids is determined as the tide height of the horizontal band within the sampling window at which muscle biomass falls below and never reattains a muscle cover of 50%. So that was how we actually decided what the position was of the boundary within a sampling grid. Observational units in linear models evaluating different independent variables' relationship to clumpy were taken to be median values of sampling window clumpy across simulated beds for given tide height values, given depth values, giving bed-wide summaries of the phenomenon of boundary formation. So these are some of the questions I wanted to answer with this model. First of all, can I reproduce a boundary, and is it robust to different parameter values? Secondly, is there evidence that protection acts as the kind of positive feedback potentially needed for maintenance of a boundary, which is what was found by Robles et al. In a cellular automata model of these muscles, and was also found in the field with Gukenzia salt marsh muscles. And finally, how does variance in a state, in this case in biomass, vary through space towards a critical transition? So these slides just walk through my field image analysis and image J, which is just an exploratory analysis to gauge the accuracy of the model in reproducing certain clumpy values. And these observed clumpy values are larger than model-generated clumpy values, something Robles et al. Also observed. The model-generated values are consistently above 0, so more clumped than not clumped, but they're not in this higher 0.6 to 1 range. So one thing that I looked at initially was how well my model captured realistic population age size structure. And this comes up in a book chapter by Donaldson, Robles, and others in 2004 that looked at different approaches to modeling a muscle bed. And so one point they made was that simpler models that ostensibly don't model certain aspects of a system do actually have implicit assumptions of what those parts of the system are. So they contrasted a 2D agent-based model in discrete space, where one muscle occupies one cell, with a 2D model in continuous space, both with absolute limits on size at 200 millimeters. And so they observe, as I do in my 3D model, that continuous space allows recruitment to continue and a more right-skewed realistic age structure to emerge. And so age structure in my model then depends not on limitations of the model, but on choices that the modeler makes about recruitment and mortality. So here are the results of my linear models for median clumpy as an outcome for different two-level parameter factors. Because this is an ABM, it doesn't have a solution that I can solve, and I have to analyze my data as though they were from a field system. So as you can see, none of these parameters have very large effects on clumpy. Protection has little or no effect on its own. Protection and predation seem to have an interaction, which is what we might expect. When there's predation, boundary intensity is higher, but that's mediated by protection. In these data, that interaction is probably mostly driven by one data point, but I've shown the same effect with other sets of output data. So here are some heat maps of muscle density with heat for a cell corresponding to some sizes of muscles in or on top of each other in that cell. We do see a very apparent difference in where the boundary forms for high and low seastar desiccation conditions. So when the seastars, even at the bottom of the bed, have a strong preference for moving downward, the boundary is lower and also, as we saw on the previous slide, more clumpy. Desiccation high versus low also has an interaction with the protection 0,1 dummy variable. So there's obviously a lot of ways in which both the model and my use of it and my analysis of the output could be improved, and one of those would be to use wider ranges of parameter values so as to be able to better understand functional forms of interactions between variables like protection and desiccation. So here's the effect of a disturbance over the 2 years it takes for the disturbed cells to recover their ability to support recruits. After 2 years, many cells are continuously being shorn of muscles and are still empty, though large muscles entering from outside and or a few muscles escaping predation could lead to reestablishment of positive feedback, maintaining high muscle cover eventually. One other thing I looked at was variance in biomass at a given depth through time. So Scheffer et al. Suggests that as a system is undergoing gradual parameter change, approaching a tipping point that's leading to a regime shift, system dynamics following any given perturbation are going to spend more time away from equilibrium conditions and therefore be less predictable. So increasing trends in variance might be early warning signals of regime shifts through space and time. However, Budiger and Hastings point out that this expectation of higher variance only holds when some assumptions about the system are fulfilled, some of which are fulfilled here, but we don't know necessarily that this system is following whatever saddle node bifurcation is theoretically required, where there's annihilation of one stable state and movement of the system to a new alternative stable state. And when we look at temporal variance in this model, variance of biomass at a point through time, if we look at this bottom row of plots with data points colored for different runs of the model, we don't see variance highest at the boundary. So in contrast to what we see with the overall system variation, we find that the non-parametric modeling approach by Carpenter and Brock that partitions variance in trends in time, or in our case spatial series, into contributions from diffusion or from rarer jumps, does detect an increase in jump intensity at the boundary. So our model suggests that this metric might be effective at anticipating regime shifts despite the presence of stochastic perturbations in nature, because the model can recognize these perturbations as the diffusion component to variance. So thanks so much to all those listed here for their support and their contribution to this project. Thanks so much for listening, and thanks in advance for any and all questions and comments.